Okay, great. Yeah, so thank you for asking me to share my research, Kelly. I added Amanda here as a co-author because I'm going to mention a little bit about her dissertation, and Amanda has really brought this powerhouse of root ecology to my lab. So I want to just give a little overview of my research on linkages between tropical forest carbon storage and fine roots with a focus on global change. And I'll talk a little bit about our long-term drying experiment in Panama, which is called PARCHED, which is the best acronym ever, which an undergrad intern came up with, Panama Rainforest Changes with Experimental Drying. And I also want to acknowledge Lee Dietrich, who's in the front of this picture, who did a lot of the groundwork, I would say 99% of the groundwork, to set this up in Panama while I was pregnant. So I wanted to start showing you guys this latitudinal graph of carbon storage across natural ecosystems. I know many of you are familiar with this, but it's a nice reminder. We know above ground carbon stocks are largest in tropical forests globally, but many people don't realize that tropical forests also hold the second largest global soil carbon stocks, second only to the northern permafrost soils, which get a lot of press attention for their carbon. We have about twice as much carbon below ground as above ground in our tropical forests. And this is sort of my main interest really is soil carbon biogeochemistry. This is what I've been studying the longest, thinking about how can we increase and protect these giant carbon banks in tropical forests. But the main input to these carbon banks, especially in tropical forests, is through roots. Root mortality and root turnover have been shown to be probably much more important than litter fall and even above ground mortality. Because decomposition rates at the surface are so fast, the roots play a huge role in feeding these giant carbon stocks. So I've actually been studying roots since my dissertation work, which was in Puerto Rico, looking at nitrogen fertilization effects on soil carbon. It was sort of the first place that I was hand sorting and looking at roots. So the overarching question of my research has been what drives variation in soil carbon cycling across tropical landscapes and with global change. And sort of the short answer to this is roots. So that's why I was here. That's why I started sort of really focusing in on roots with Amanda. So as I said, I started my research in Puerto Rico. I did a postdoc then in Hawaii, looking more at fire effects on soil carbon storage and invasive species. But for the past 12 years or so, I've really been focused on the lowland forests of Panama. So here we see the Isthmus of Panama, and all of those dots on there are long-term 1 hectare forest dynamics plots, which have been maintained by the Smithsonian Tropical Research Institute, where I'm a research associate. And these forest dynamics plots have had a lot of long-term research on them. And actually the Barrio Colorado Nature Monument, we just had our 100-year anniversary of research. But before I started working there hadn't been a lot of work on soil carbon, which was sort of surprising and very lucky for me. So one of the first things I did when I started working in Panama was this large-scale biogeographic study of what are the soil carbon stocks out there, how much carbon is there, and how does it vary across different forests. So what you see in the background of this map is the geological variation. And Panama has unusually high geological variation over relatively short spatial scales. And this is largely because this is where the northern and southern hemispheres came together 3 million years ago, which led to the great migration of species north and south, and also gave rise to all of this geological variation, which is really well correlated with changes in soil fertility now. So we have 2 orders of magnitude of variation in soil phosphorus and base cations across really small spatial scales in Panama. And we also have a doubling of rainfall from the southern Atlantic to the northern Caribbean. And that's not correlated with soil fertility, which is somewhat unusual and allows us to sort of disentangle fertility and rainfall effects. So the main takeaway from that first study is shown in the different colors of these dots, which show sort of low, medium, and high levels of carbon in yellow, white, and black. And I'll point out that even our low levels, our lowest levels are still about 7 kilograms of carbon per meter squared to 1 meter depth. And in a global sense, that's very high carbon stock. But for us, that's sort of low. And you can see here that the carbon stocks really don't change in a noticeable way across the rainfall gradient. It's much more of a hopscotch. And so this led to subsequent questions about, okay, well then what is driving soil carbon storage? So before I answer that question, though, I just want to give you a sense of the variation and diversity of soils that we have. So we have 5 of the 12 USDA soil taxonomy soil orders in Panama across the lowlands. Everything from our typical strongly weathered red oxasols, like you see here in the upper left, to tropical mollisols, which are less common below that, alphasols, ultisols, and even inceptisols. Although our inceptisols don't fall neatly into the USDA bin, because often they're made from deposition of strongly weathered clays. So they're strange inceptisols. Okay, so going back to this question about what's driving soil organic carbon, we measured over 30 ecosystem parameters to see what would explain that, including above ground biomass, above ground productivity, litter fall, all the soil nutrients, soil toxicity, texture, et cetera, et cetera, et cetera. And using structural equation modeling, this was the most parsimonious predictor of soil organic carbon. And you can see the strongest direct predictor is fine root biomass. This is live biomass. So a really coarse scale measure of fine roots for this group. And then the fine roots in turn were inversely predicted by total extractable bases. So the more infertile the soil, the larger the root biomass stocks, as we would expect because of sort of nutrient scavenging in soils. And then those together really strongly predicted organic carbon. We also had an effect of soil clay content. So clay is typically positively correlated with carbon because of sorption potential on clay surfaces. And then a weak but positive signal of rainfall, which as you saw isn't so easy to see when you map it all out. So after this, right around this time, I also got a Department of Energy Early Career Award, which was really focused on this question about the roots. And so Rich Norby found me at a national meeting in Washington, D.C. right after I got that grant. And he's like, hey, you don't know that much about root ecology, right? And I didn't know Rich at the time, but I was like, oh, I do. I know enough about it. And he's like, yeah. Okay, well, there's this lady you should really meet, Amanda Longi Cordero. She's finishing her master's in Brazil, and I think you really need her to get this grant going. And boy, was Rich right. So luckily, Amanda came and joined my group and brought a much broader perspective beyond biomass, which is what I had been really focused on. And so we've been doing a lot of other work, which I'll just mention briefly. Sort of early on in that, we looked at fine root depth distributions to see how those corresponded to carbon storage. And really sort of a simple version of all of those 50 sites is shown here. Infertile sites are on the top panels. Let me see if I can get my pointer. So we have all of the infertile of those 50 sites sort of grouped up here. Here are fine roots. We see this typical depth distribution where we have less roots at depth. And that was inversely related to the distribution of carbon depth. So a lot of roots in the surface of infertile soils, apparently inputting carbon that's getting vertically transferred and then sorbed in deep soils. And then we see sort of a dampened signal of this in the fertile sites. And in between these, there's a whole gradient of intermediate fertility that I'm not showing. So this has all been sort of thinking about root biomass, which as Dani and Lainara show in their review paper is what we have most of the data on biomass. But with Amanda, we're interested in looking at root morphology, other traits, how are those related to carbon storage? So here is a nice figure that Amanda has in her dissertation, which is about to be finished in the next few months. So I felt comfortable showing this. This is a paper that's going to be submitted hopefully in the next few weeks. And so what Amanda did first is show this really nice positive correlation with fine root biomass. This is sort of recreating from independent data, that trend that I showed you from my data. But then she did all of this beautiful root morphology and root chemistry and showed this interesting negative relationship between root-specific root length, fine root-specific root length, and total carbon. So the longer and thinner roots are corresponding to less retention of carbon in soils, which makes intuitive sense because of the tissue type, right? Long, thin roots, we think maybe they would be decomposed more easily and lost from soils. And then she showed the same thing for extractable organic carbon, an independent measure of soil carbon. So we have this really nice relationship between root traits and total carbon. And this kind of data I don't think is out there at all for tropical forests. So I'm excited for her to publish this. As I mentioned, so all these things I've been talking about are this sort of large-scale biogeographical observational studies trying to explain what's happening out there on tropical landscapes. But we're also interested, as I said, in global change, especially drought and drying. So right now, Panama is undergoing one of the worst droughts in its history. The canal is so low that boat traffic is being diverted. They're not able to actually bring boats across the canal. And all of our sites sit right along the Panama Canal. So drying is a big deal in Panama. And here is another picture of that through fall exclusion experiment that Lee helped to set up. We were excluding 50% of through fall, and then a couple years ago, we increased that to 70% to enact a stronger drought effect. And so we really wanted to do this because across the natural rainfall gradient, we saw a weak but significant influence of rainfall on carbon cycling. But we want to know, well, what happens if you really dry down these forests in situ, right? All those forests across the rainfall gradient are adapted to their background rainfall levels. But what if we enact these big chronic drying events? So I'm not going to go into all of the data from this. We've had a few great papers come out in the last year or two from Lee, from a soil microbiologist, Stephanie Chacon, and Nick Buskill. Lee and I, with Ben Sulman, who's a soil biogeochemical modeler, who's now at Oak Ridge, we recently published a paper looking at soil carbon losses through respiration with drying. And we've seen some divergent effects, which seem to be related to soil fertility. So it's really interesting. So how these forests are holding on to carbon seems to be changing with drying. And then I'll just put one more figure from Amanda's dissertation, which is, as I said, pending. And Amanda did a lot of nice work on root production and root turnover using both ingrowth cores as well as mini rhizotron imaging. And what her data are showing is that this in situ dry down is reducing fine root turnover and also reducing fine root productivity. So this is sort of initial data for that, showing that we're really affecting root growth and inputs to the carbon stocks. And then together with the data from Lee and I, that we're also changing the soil carbon losses. So we're starting to put all of this together into sort of larger soil carbon models. This is sort of how we think about these models. From my perspective, I just really focus on the carbon, sort of the mass balance of carbon coming into different soils from root biomass and turnover, as well as litter fall, which I said earlier is less important. And then the balance of that with soil respiration, which is our main loss. Here are two of our soils that we sort of use as the poster children of high soil carbon stock and oxisol on BCI, and then lower soil carbon stock and alpha-sol that's at a different location in the Barro Colorado Nature Monument. So in addition to this mass balance, though, especially with Amanda's work, we're really trying to bring in some more nuanced view of roots and what root tissues do and root chemistry, so the role of morphology and turnover rates. So I think we're really going to be able to improve this sort of modeling mass balance approach by including more of this sort of nuance. And so we just found out at New Year's that we got an NSF LTREB grant, which is the 10 years of funding to keep this experiment going. And so that was focused, sort of the reason for that was because of all the interannual variation and the increase of extreme droughts. So we have 10 more years to keep looking at this. Unfortunately, Amanda's graduating, but I have some more students coming in next year. And anybody who's interested in drying effects on tropical roots, I would love to have more collaborators at these sites. So I'll end there. I'll acknowledge part of the massive team that helped with this, the huge field team, which is not even a complete list, but I couldn't fit more names on here, the lab team and many collaborators who've worked on this, especially across the national lab system in the US. So we'd love to have more international collaborators.